What if I told you that scientists have just cracked the code to edit genes in adult brain cells with unprecedented precision? Something that was considered nearly impossible just years ago? This breakthrough could fundamentally change how we approach brain research and potentially even treat neurological diseases. But here's where it gets controversial. While traditional gene editing methods have been struggling in mature brain cells for decades, this new approach essentially tricks the cell's own repair machinery into doing exactly what scientists want. Some might argue we're playing with fire when we manipulate the brain's genetic blueprint so precisely. Researchers have developed a revolutionary technique that transforms the landscape of genetic engineering in cells that have stopped dividing, particularly adult brain neurons. Their secret weapon? An ingenious design using microhomology tandem repeat repair arms that simultaneously activates two different cellular repair mechanisms that scientists previously found difficult to harness together. Let's break this down in simple terms. Think of your cell's DNA repair system like a toolbox with different tools for different jobs. Most gene editing attempts in mature brain cells rely on just one tool, non-homologous end joining, NHEJ, which often creates messy, imprecise results. It's like trying to perform surgery with only a hammer when you really need a scalpel too. Here's the game-changing part. These researchers figured out how to use both the hammer, NHEJ, and the scalpel, microhomology-mediated end joining, or MMEJ, at the same time. The result? They achieved editing success rates that are 5 times higher for maintaining proper gene function, and doubled their success for clean, scar-free genetic modifications. Traditionally, scientists have hit a massive roadblock when trying to tag genes in brain cells. Unlike rapidly dividing cells that can easily repair and modify their DNA, mature neurons are essentially locked down. Their standard DNA repair highway, called homology-directed repair, HDR, becomes virtually unusable. This limitation has frustrated neuroscientists for years, preventing them from easily studying how proteins behave in living brain tissue. And this is the part most people miss. The researchers didn't just stumble upon this solution. They strategically designed special DNA sequences called frame-retentive microhomology tandem repeats. These sequences act like molecular GPS coordinates, guiding the cell's repair machinery to make precise, predictable changes rather than random, potentially harmful modifications. To test their revolutionary approach, the team targeted a specific neuronal gene called TUB2A, which plays a crucial role in maintaining the internal skeleton of brain cells. They used a sophisticated dual virus delivery system. Imagine it as a 2-part molecular package deal. One virus carried the genetic scissors, Cas9, while the other delivered the instruction manual, guide RNA, and the new genetic material they wanted to insert, including a green fluorescent protein tag that would light up successfully modified cells. The results were nothing short of spectacular. 3 weeks after treatment, brain tissue examination revealed bright green fluorescence, specifically in neurons from the treated areas, proving that the editing had worked and the cells were producing the modified protein from their own genetic instructions. Even more impressive, advanced whole brain imaging techniques revealed the green-tagged proteins distributed throughout extensive neuronal networks in both cortical and hippocampal brain regions. But here's where it gets really fascinating and slightly controversial. When the researchers performed detailed molecular analysis to verify their results, they discovered something that challenges conventional thinking about DNA repair in brain cells. While traditional NHEJ-based editing accounted for only 1.8% of successful modifications, Their microhomology-enhanced approach achieved an impressive 8.6% success rate through the MMEJ pathway. Even more remarkable, the MMEJ repairs followed an incredibly predictable pattern, a consistent 6-nucleotide deletion that perfectly connected the new genetic material with the existing gene. This level of predictability was actually forecasted by artificial intelligence algorithms, specifically a deep learning platform called inDelphi, demonstrating how computational biology is revolutionizing experimental design. The implications of this breakthrough extend far beyond simply tagging proteins with fluorescent markers. By enabling reliable, precise genetic modifications in adult brain cells, this technology opens unprecedented possibilities for studying neurological diseases, mapping protein interactions in real time, and potentially even correcting disease-causing genetic mutations directly in the brain. What makes this approach particularly exciting, and perhaps controversial, is its versatility. The underlying principle of exploiting microhomologies to simultaneously engage multiple DNA repair pathways could potentially be applied across different cell types and even different species. We're talking about a fundamental shift in how we approach genetic engineering in mature, non-dividing tissues. The integration of artificial intelligence prediction tools proved crucial to the method's success. This deep learning-assisted approach allowed researchers to anticipate how cells would respond to their genetic modifications, enabling them to design repair templates that consistently maintained proper protein function. This represents a perfect example of how cutting-edge computational biology and experimental techniques are converging to solve previously intractable problems. The biological relevance of these modifications was beautifully demonstrated through whole brain imaging, which showed that the fluorescent-tagged proteins were correctly localized within neuronal structures, confirming that the approach produces functional fusion proteins that accurately reflect natural expression patterns. Beyond neuroscience applications, the concept of frame-retentive microhomology tandem repeat repair arms engaging dual DNA repair pathways has profound implications for gene therapy, synthetic biology, and the development of programmable cellular systems. The persistent challenge of improving efficiency and accuracy in genetic modifications of slowly dividing or non-dividing cells has long been a critical barrier for clinical genome engineering. And this method offers a promising solution: the practical use of adeno-associated viruses, AAVs, for delivery adds another layer of clinical relevance, since AAVs are already proven FDA-approved vectors for human gene therapy applications. By eliminating dependence on HDR-based repair mechanisms, this approach could significantly enhance the applicability of in vivo genome editing strategies in mature human tissues, including potentially the human brain. Perhaps most importantly, this research challenges long-held assumptions about the limited DNA repair capabilities of mature neurons. It reveals that these cells possess a more flexible and accessible repertoire of repair mechanisms than previously thought, opening new possibilities for therapeutic exploitation. This study represents a masterclass in how precise molecular design, informed by computational prediction and supported by elegant experimental validation, can dramatically enhance genome editing efficiency and predictability. It establishes a new gold standard for endogenous gene tagging and creates a roadmap for future genetic interventions in brain tissue and other non-dividing cell populations. By successfully engaging both NHEJ and MMEJ pathways through intelligent template design, these researchers have transcended traditional limitations of genome editing efficiency in neurons. This breakthrough could catalyze applications ranging from fundamental neuroscience discoveries to clinical gene therapies, perfectly illustrating the power of combining synthetic biology with computational foresight. The ability to generate scar-free, predictable, and functionally relevant genetic integrations in differentiated cells may indeed herald a new era in genetic engineering, potentially accelerating discoveries and treatments that were previously constrained by technical limitations. Here's my question for you. If we can now precisely edit genes in adult brain cells, should there be limits on how far we push this technology? Are we crossing ethical boundaries when we can potentially modify human cognition or memory at the genetic level? What safeguards do you think should be in place as this technology advances toward clinical applications? I'd love to hear your thoughts on whether this represents human progress or a step too far into uncharted territory.